Hello, my name is HumeStroud. I'm an assistant professor at the University of Texas Southwestern in Dallas, Texas. I appreciate the opportunity to give you an overview about some of our work. Our lab studies epigenetic control of gene expression in the brain and seeks to determine how mutations in these epigenetic factors give rise to neurological disorders. Today, I'm going to focus on one of our projects that involves methyl DNA binding protein 2, or MECP2. Heterozygous loss-of-function mutations in the X-linked MECP2 gene causes Rett syndrome, a really devastating neurological disorder that primarily affects females. Girls with Rett syndrome appear to develop normally for the first year or so, where they typically learn to walk and speak their first words. However, after this developmental period, they progressively exhibit loss of speech and motor skills. They also display a wide variety of neurological defects, such as seizures and cognitive impairments. MECP2 mutations in mice recapitulate Rett-like defects in neural circuits and behavior, allowing us to study this disorder in mice. Studies suggest that this disorder is reversible in mice and opens the possibility that Rett syndrome might be reversible in humans as well. Today, I'm going to focus on one question, which is where does MECP2 bind in the genome? Because MECP2 is a DNA binding protein, understanding where in the genome MECP2 binds to will provide insights into the function of MECP2. MECP2 was initially found 30 years ago by Adrian Bird's lab as a protein that binds to methylated DNA. Cytosine methylation is a key covalent modification that occurs across the genome. In most mammalian genomes, methylation occurs exclusively in CG sequences. So these are cytosines that are followed by guanines. And CG methylation at the promoters of genes is classically associated with gene silencing. And it was found that MECP2 tightly binds to CG methylation. With Huda-Zoghbi's discovery that mutations in MECP2 causes Rett syndrome, many groups have since studied the roles of MECP2 in gene regulation. But historically, there's been very limited consensus on the mechanisms by which MECP2 regulates genes. About 10 years ago, the labs of Bing Ren and Joe Ecker discovered that neuronal genomes have extremely high levels of non-canonical DNA methylation that occurs as CA sequences. So these are cytosines that are followed by adenines. In human brains, the amount of CA methylation can uh, exceed the amount of canonical CG methylation, suggesting that in our brains, the dominant form of methylation is actually CA methylation. Soon after the discovery of high levels of CA methylation in the brain, the labs of Hongjing Song, Mike Greenberg, Huda Zoghbi, Adrian Bird independently found that MECP2 tightly binds to CA methylation in vitro and in vivo. So once bound to CG and CA methylation, MECP2 appears to function to restrain the transcription of many key neuronal genes across the genome. This is great as it seemed like for the first time almost that multiple groups agreed on a mechanism by which MECP2 mediates gene regulation. Although methylation-mediated gene regulation is definitely a primary way by which MECP2 regulates genes, genetic evidence suggests that MECP2 likely regulates genes independent on methylation as well. I think a major challenge on studying MECP2 has been because it seems to be bound everywhere across the genome. Typically, when we want to study a transcription factor, we identify which genes that the transcription factor binds to and regulates. For example, in this case, using the Krebs binding protein, or CBP, it is clear which genes this CBP binds to. So the peaks here represents CBP binding sites, and you can see ascribe which genes are bound to and regulated by CBP. By contrast, if we look at similar data for MECP2, what you can see is that it seems to be bound in the genome pretty uniformly without exhibiting peaks like CBP. So I think this is one of the factors that have made studying MECP2 difficult, as target genes were somewhat unclear. And most previous assays that have mapped MECP2 were done with cross-linking and sonication approaches, including the data shown here. So we decided to reinvestigate MECP2 binding to the neural genome by using a different approach called cut and run, an assay that enables sensitive quantification of protein DNA binding across the genome in situ. So, in addition to confirming that MECP2 binds to methylated DNA as reported by previous studies using different approaches, surprisingly, we noticed that MECP2 cut and run forms these discrete peaks enriched at specific sites. This is just showing you one genomic locus of the GABA receptor gene that is known to be misregulated in MECP2 knockout. And what you can see is that there are focal enrichments of MECP2 across the gene. Importantly, these focal enrichments or peaks of MECP2 are abolished when MECP2 is deleted, suggesting that these binding sites represent MECP2 binding. IgG controls from the same samples processed in parallel do not exhibit such peaks, indicating that the signal is specific to MECP2. This pattern of MECP2 binding is quite different from what others have reported over the years, and this is a somewhat strange result, but we did do a series of control experiments and think that they're not artifacts of the assay. So by using multiple biological replicate, we identified 20,000 such peaks and named these sites MECP2 binding hotspots, or MBH sites. Because MECP2 is thought to bind to DNA methylation, we first thought that MBH sites might be marked by DNA methylation, which will then recruit MECP2 to MBH sites. To test this, we perform whole genome bisulfite sequencing, a method that allows for quantification of DNA methylation at single base resolution across the genome. And what we found through this experiment was surprisingly that MBH sites were depleted in DNA methylation. Here we aligned all 20,000 MBH sites at the center and plotted the average distribution of CG methylation at the top and CA methylation at the bottom, and plotted the distribution of methylation across the MBH sites. And what you can see is that both CG methylation and CA methylation tends to be depleted at the MBH sites compared to the flanking regions. So this suggests that MBH sites are depleted in DNA methylation, suggesting that MECP2 may not bind methylation. In data I don't have time to talk about today, we further found that reducing methylation in vivo does not reduce MECP2 binding at these sites, suggesting that MECP2 interacts with these MBH sites independently of methylation. So what it seems like is that MECP2 interacts with MBH sites independent of DNA methylation. There appears to be distinct classes of MECP2 binding sites. One is more traditional MECP2 binding to CG or CA methylation, but there's also these very specific sites where MECP2 is highly enriched, where MECP2 appears to bind independent of DNA methylation. So to begin to understand the function of these MBH sites, we wanted to see where in the genome these sites are located. And what we found was that MBH sites were mostly located at distal gene enhancers, which are key gene regulatory elements that are known to modulate gene transcription. So this suggests that MBHs may regulate enhancer activity. Because MBH sites were located at gene enhancers, we asked whether MBHs are located at genes that are regulated by MeCP2 or known to be regulated by MeCP2. And what we found was that MBH sites were significantly associated with genes that are repressed by MeCP2. By contrast, MBHs were not associated with genes that are activated by MeCP2, suggesting that MBHs may primarily mediate gene repression. A striking feature we notice is that the more MBHs near the gene, the more repressed the gene becomes by MeCP2. Here on the x-axis, genes were ranked based on the number of MBHs near the gene in wild type, and on the y-axis is the extent of gene repression by MeCP2 for these genes. And what you can see is that the more MBH sites the gene has in wild type, the more repressed the genes are by MeCP2. This suggests that MeCP2 preferentially represses genes harboring multiple MBHs, suggesting potentially a new mode of gene regulation by MeCP2. And uh, what we've noted is that quite often these MBH sites are present in broad clusters, as shown in this one example here of the ADGRL2 gene that is strongly repressed by MeCP2. We identify a few hundred of such broad clusters. So it seems like genes that are bound by many MECP MBH sites are particularly strongly repressed by MECP2. So to summarize our findings, we discover a novel class of MECP2 binding sites that we named MBH sites. These were not found or detected by other studies. Uh, we surprisingly find that MECP2 binds to these MBH sites independently of DNA methylation, suggesting a methylation-independent mechanism by which MECP2 regulates genes. And interestingly, MECP2 appears to repress genes harboring multiple MBH sites. So uh, if a gene has one MBH site, it's less repressed than a gene that has 10 MBH sites. So we think this is a new way by which MECP2 may regulate gene expression. So I think our study ended up opening more questions than we successfully answered, and there are many questions that remain. One thing we're very interested in is understanding or defining the interaction between MECP2 and MBH sites. We don't yet know how MECP2 is recruited to MBHs. It could be a direct interaction or indirect interaction. So we want to determine the DNA sequence and chromatin features that within MBHs that MECP2 recognizes in vitro and in vivo. Because MECP2 has multiple domains across the protein, we want to identify the domains that mediate this specific binding of MECP2 to MBH sites. And finally, we like to assess the impact of Rett syndrome, human Rett syndrome patient mutations on MECP2 binding to MBH sites. Finding that specific disease mutations disrupt MECP2 binding to MBH sites will underscore the functional relevance of MBH sites to Rett syndrome pathology. We're also very interested in understanding mechanisms by which MBHs represses transcription. We don't yet know how MECP2 regulates genes. And what we're interested in is understanding how MECP2 and MBHs control enhancer activity and how it mediates this gene repression. So to do this, we want to identify the interactors of MECP2 selectively at MBH sites. And by doing this, we want to study how MECP2 MBHs and these potential interactors collaborate to repress gene transcription. Our hope is that these studies will advance our understanding on the mechanisms by which MECP2 regulates neuronal gene transcription, and it also provide much needed insights into the etiology of Rett syndrome. With that, I'd like to thank my lab members, in particular Guillaume Mishra, a postdoc who has led this work, as well as Eric Sun, Tiffany Chen, and Mandy Eckhart, who have supported this study, as well as my collaborator and former mentor, Dr. Mike Greenberg. And thank you all for your attention.